This is Joe narrating a PowerPoint on page 518 in the Kaplan book. It's lesson 6, Life Science, Modern Genetics. Key ideas are that the information that every cell contains is contained in the DNA molecule within the nucleus of the cell. Humans have 23 pairs of chromosomes. If you were to stretch out this DNA molecule contained in each nucleus of each cell, it would stretch out to be 6 feet long. Along this molecule of DNA, there are locations on the molecule that are called genes. Each gene usually makes a specific protein. Your body is made of proteins, things like your hair, your fingernails, the organs in your body, and also the proteins that perform the chemical reactions in your body are called enzymes, and these are also made by the information within the DNA. There are many genetic diseases, and we are at the threshold of altering human cells to make them free of genetic diseases using new techniques. This first slide shows a cell. From that cell, we take a closer look at the nucleus. The nucleus contains, in humans, 23 pairs of chromosomes. And this chromosome, each chromosome is one long molecule of DNA. The information in the DNA is coded by a series of 4 different chemicals: adenine, thymidine, guanine, and cytidine, and these are abbreviated as A, T, G, and C. So the order of these chemicals on the DNA molecule specify information. This information in the DNA is translated into a different type of nucleic acid called RNA. This RNA then leaves the nucleus. It goes out into the cytoplasm of the cell, and at that point the information now contained in the RNA is transcribed or written as a series of amino acids. Amino acids are the molecules that make up a protein. These proteins, as said, make up the structure of the body and also perform chemical reactions in the body. So just as the English language has letters, these letters can be put in different orders to make words, and then these words can transmit information. The 4 letters A, T, G, and C in DNA can be translated into the information that allows the cell to make proteins. The genetic code is the basically the translation of each 3-letter segment in the DNA into a specific amino acid. So for example, on the left-hand side you see the first letter of the triplet code, either U, C, A, and G, and U is uracil. That's the nucleotide or molecule that's in RNA. At the top you see the second letter, and on the right you see the third letter. So you can combine these 3 molecules to make a 3-letter code that then gets translated into a specific amino acid. For example, if you look at the upper left, if the RNA has UUU together, then that triplet will become the amino acid phenylalanine. So in this way, the information in the series of letters in the DNA becomes a series of amino acids in a protein. So this is what's called a karyotype. That's K-A-R-Y-O-T-Y-P-E. It's a display of the 23 pairs of chromosomes in a human cell. This cell happens to be from a male. As you can see on the lower right, this cell contained an X chromosome and a Y chromosome, which marks it as a cell from a male. So these 23 pairs of chromosomes contain all the information for that cell, and half of these chromosomes will be transmitted to the next generation. meaning half from the mother and half from the father. We can look at the actual DNA sequence now in human cells, which basically means taking a look at these chromosomes with very high magnification, so we can actually see the letters A, T, G, and C that go from one end of a chromosome to the other. We've done this for all 23 pairs of chromosomes in the humans, and this is called the human genome. Once you have the order of the letters in a human cell, you can manipulate those letters in the DNA and hopefully correct genetic disorders. So we are on the threshold of being able to do that. Take a single cell, alter the sequence of letters in the DNA that would then correct a protein being made in the wrong way, and then would correct a actual human disease. So that's the end of this, Modern Genetics, on page 518 in Kaplan.